Okay guys, so we're going to go through the last lab of the semester. Hooray, you're almost done. Congrats that you made it this far. Great job. So this lab is about the gram-negative bacteria that are normal microflora of the GI system and can become opportunistic pathogens if the case arises. So some of these are aerobic, some are facultatively anaerobic, some produce gas, some utilize lactose, some don't. And the tests that are done on the gram-negatives are different than the gram-positives, and they're a little bit more complex. So here's a couple that are commonly tested for in laboratory settings because these are the ones that are typically occurring in humans. And so this chart should look semi-familiar. There was a similar one for the gram-positive identification lab where you differentiated staph from strep. In this virtual lab, you're going to be identifying two different unknown gram-negatives. And I believe they are from the bottom part of this chart. Okay, so the gram-negative bacilli. And there are fewer branching points, so there are fewer tests. However, you'll notice that at the end of some of these branches, there are 2 microbes or 3 microbes listed. So we have to find a way to differentiate between E. coli and Klebsiella, for example. So essentially, there is a special test that we can use to do that. So it takes into account multiple other types of tests that we'll talk about. So again, normal flora in the gut, Escherichia coli, Proteus, Enterobacter, Klebsiella, some of these you guys have already seen in lab, and then the type of infections that they cause are shown as well. So this is just a reminder of what some of those microbes looked like on the EMB agar plates. And some microbes can be identified visually by looking at how they grow or the colors that they show. Remember, we did that a little bit in lab using the colony formations. However, if you remember, some of them have similar characteristics, so it makes it hard to use visual characteristics alone to differentiate between 2 different microorganisms. One of the few that does have very distinguishing characteristics is Proteus. So it forms these concentric rings or swarms on agar, distinguishing it easily from other microbes. Pseudomonas is another one that has very distinct characteristics when cultured on certain types of agar, like tryptic soy, where it shows a blue-green color. There's a few other things listed here about it for you to read. It is a common hospital-acquired infection, which is why we study it. So gram-negatives, we have to test for certainty using biochemical testing. Because again, as you remember, looking back at that chart, there are a couple possible gram-negatives that could be identified from one branch of the gram-negative rod part of the tree, the identification tree. So what we do is we use a biochemical test to look for the presence of certain chemicals or enzymes or proteins. And if those things are present, it will cause a color change in the media. So the 2 different types of tests that can do this are the IMVIC test and the Enterotube test. Some of the IMVIC tests are also part of the Enterotube test, which is why that's the one we typically do. So this is an example of the IMVIC breakdown. So the things that are tested for are indole, methyl red, Voges-Proskauer, and citrate. And again, these cause a color change if the enzyme in question is present. So indole changes color like purple if tryptophanase is present because it converts tryptophan to indole. So you're essentially testing for the metabolite of the enzyme, which is true for most of these. And then the other 2 are combined, the methyl red and Voges-Proskauer, and that identifies the type of glucose metabolism occurring. Okay, and then the citrate test just identifies whether or not the microorganism can utilize citrate as the sole source of carbon. If you remember, that's part of the citric acid cycle. It's green if it's negative, and it's a blue color if it's positive. And you can watch a little video about each of these tests combined, or the IMVEC test, but you won't need to be worrying about it for your part of the assignment. So this is a cartoon version of the Enterotube test, and there's a nice YouTube video explaining some of these tests and how you inoculate an Enterotube. And it's worth a watch, so I suggest you take a moment and take a look at this when you're all done with this video. So again, the way this works is there are multiple chambers of agar containing some kind of indicator that will change color if the microbe contains either lysol or indole, if it can ferment lactose, etc., or if it can utilize citrate. So if the microbe can use or contains one of these substances listed across the top, then a color change will occur. So you'll notice that there are many possible combinations that can occur for each microbe. So we use a chart to identify the microbe based on the different number of positive tests, and you'll see that in a moment. So here's an actual picture of the Enterotube. The bottom is the uninoculated control, and the three on the top are 3 known organisms. So Proteus vulgaris, Enterobacter, and Escherichia coli, or Escherichia coli. And then again, numbered across the top are the different tests. So this is just to show you examples of what the color changes would look like for a positive test. So spend some time taking a look and comparing the different colors from the top 3 inoculated enterotubes against the bottom uninoculated tube. And so here's just another explanation. Take your time, read through this, of the uninoculated tube on the top and positive tubes on the bottom, or positive tests on the bottom. So some microbes, depending on their metabolism, can produce a gas as a byproduct. Remember, fermenters can produce this, like alcohol fermentation, and microorganisms that do that release the gas CO2. And so the way that's captured is with a waxy substance that's placed over top of the agar portion of the chamber. And if the species releases gas as part of its metabolism, it will be released from the agar and push the wax up. And you'll see this gap like you do in the bottom of the picture next to the bracket. Okay? So I think I tell you if there's gas produced in the picture, because it's really hard for you to see that in your virtual lab. So why do we study this? Enterics are the most common cause of UTIs and other infections of the lower GI. And there's a special streaking pattern done for urine cultures. You would normally do this in lab. That's why we're talking about it briefly. And this is what it looks like. It's kind of straight down the center streak, and then you kind of, like, instead of doing a 4-quadrant streak, you just streak it back and forth from top to bottom after you've done the one streak down the middle. And then in some cases, there are dual plates that have 2 different agar, so that you can perform more tests at one time. Okay. Other tests done on urine for identification purposes, aside from culturing, is just microscopic analysis to see if there are any protists or fungi present in the urine. So for what you actually have to do for this lab, you need to use the chart on the next slide and identify the organisms. There are 2 inoculated in these 2 tubes. One error on the slide showing is that it says the bottom tube is uninoculated. That's not correct. It's on the next slide, or the few slides previous that showed the 3 inoculated tubes and the 1 uninoculated tube. So both tubes in this photo are inoculated, and you will have to identify the organisms. Okay. The bottom, because it's a little bit hard to tell for some of them, I did put the key for which chambers are in fact positive to help you out there. So what you're going to do, again, compare these 2 tubes to the uninoculated tube a few slides back and on the chart on the next slide. Okay, and then tell me which tests were positive and which tests were negative, and then which microorganism you had in each of these tubes. Okay, so across the tube, if there was a color change different from the uninoculated tube, then it's a positive test. Okay, positive/negative means some of the microorganisms are positive and some are negative. It depends on the strain. So it could be a positive test or it could be a negative test. Okay, so you're going to look at the tubes, determine for each of these tubes shown in the previous slide which of them had the color change, is it positive, is it negative, and compare them against this chart. And then you can tell which microbe is in which enterotube. And that's it. So you're going to submit your response to me on D2L. There'll be a submission area. Okay, if you guys have any questions, please reach out via email and I'll get back to you as soon as I can. Take care.